Obviously, living things need to ingest certain molecules in order to survive. So, we have to ingest lipids and carbohydrates and proteins, and some of them are essential in that we can't make them. There are essential amino acids, there are essential fatty acids, there are vitamins. Our bodies can't make them from other things. Unless we ingest them from other sources, then we struggle to survive. With bacteria, we could ask the same question, and as geneticists study bacteria and their mutations, this becomes important. Are bacteria prototrophs, in which they can make their own essential molecules? Their genome encodes the enzymes which can convert, say, one molecule into this essential nutrient? Or are they auxotrophs, where they lack that ability, and therefore there is a nutrient or nutrients which have to be supplied to them, otherwise they would die? And so here I'm showing 4 different bacteria, and we'll ask the question, are they prototrophs or auxotrophs? Now obviously these are 4 different species, given that they have different shapes and they're different in their gram negative or positive staining, but they could be 4 different strains of the same species as well. So if we were to do what's called replica plating, where we stamp existing colonies which are growing in agar which has enriched media like luria broth that has all of the nutrients they need. So in this upper petri dish, they can all grow because they're supplied with a rich set of nutrient supplements. If there's, say, and originally it was velvet which would be put on these stamps, if this velvet then takes up some of these bacteria and we then apply it to a second petri dish, all of the same bacteria will inoculate the second petri dish in the same places. And so then we could ask, if the second petri dish then is made with minimal media, which lacks many of these nutrients, how do these bacteria fare? And as you can see, strains 1, 2, and 4 fail to thrive. They die because they apparently need something which the medium does not include. They are what are known as auxotrophs. Only strain 3 can support itself in this minimal media agar plate without other supplements, so it then is a prototroph. If strains 1, 2, and 4 are auxotrophs, which require a specific nutrient in order to survive, which nutrient might that be? Well, we could prepare additional plates with minimal media, and we know that only strain 3 can survive on minimal medium, so the other 3 need something that minimal media does not contain. So we make 3 additional plates. One is composed of minimal media plus lysine, which is supplemented to it. The second plate is enriched with arginine, and the third plate is enriched with histidine. If bacterial strain 1 can survive on a plate with minimal media and lysine, but not on minimal media, that means that it is an auxotroph which is unable to synthesize its own lysine, and then lysine must be supplied in order for it to grow. We might, depict it as lys minus, Lys minus, meaning that it can't make its own lysine. Strain 2 would be an auxotroph, which would be unable to survive without arginine. It can survive on minimal media with arginine, but not on minimal media without arginine. So it would be Arg minus, an auxotrophic strain. Meanwhile, the 4th bacterial strain would be His minus. Apparently it can't grow with minimal media, but it can grow with minimal media and histidine. So histidine must be the metabolite that it is unable to produce. So it is an auxotroph His minus.